Okay, so first, thanks Tamar, Anton, and Jan for the very nice meeting here in Vienna. That's the first time for me. I really love the city, and I've learned many things so far. So, very great experience for me. So today I will talk about replication and cohesion. The title have a little bit changed. You will see it's related. So my group is mostly interested in chromatin regulation and chromosome organization using polymer modeling and statistical physics. And one of our long-term interests is the coupling between chromatin marks, epigenetic marks, and chromosome organization. And I mean, since we have many times for discussing, I mean, we have very recent story on the regulation of marks related to the 3D organization and the regulation of the 3D organization related to the marks. And so I'm very happy to discuss about that. But today I would like to talk about different things. So as you may know, there is two very important mechanisms that drive the 3D organization of genome. One is SMC-mediated loop extrusion, and the other is phase separation mediated by architectural proteins like HP1 or polycomb. And actually, you may see that as the effect of translocating motors for loop extrusion and the effect of condensation or attractions for the formation of compartments. And actually, there is an interesting and well-known beast in the cells, polymerases that are translocating motors along the genome, and that potentially have this capacity to self-attract. And it's well known for years, based on early microscopy image, that potentially this type of machineries can form what's called replication factories for replications or transcription factories for transcription. So basically colocalization of these translocating motors in some kind of condensate. There may have been many controversies about the reality of these factories. We'll not discuss too much about that. We recently have also a nice story on the transcription part, but today I will focus on very exciting ongoing work regarding the effect of DNA polymerase and replication on the 3D organization of chromosomes. And the hero of this work is Dario. He's here in the back, maybe here, who's really leading this project. He's a PhD student in my group, and we co-supervising him with Cedric, a long-term collaborators, and Jean-Michel, who is an expert in replication. And Dario is a poster, so you can ask more details to him. So replication is actually quite fascinating. So eukaryotic cells and replication in eukaryotic cells start from multiple, what is called origins. Basically, at multiple positions, replication starts, and then occurs along the genome. And in the early days of high-C experiments, actually, there were already a nice observation that the different compartments that you can see in the high-C map are also tightly correlated with the timing of replication. So if you are replicated early, it's mostly associated with active compartment, and the late replicated regions are mostly the B compartment. And this relation between the 3D organization and the replication timing was mostly hypothesized as a problem of accessibility, because the beginning of replication is related to firing factors that potentially may have to diffuse inside the nuclei, and so potentially, okay, the structures may have an effect on the diffusion of these firing factors and may trigger the early or late firing of these events. Yes. I think it's not working. But this is still an hypothesis, okay? There is not really concrete proof of this hypothesis, but today I will try to ask the question differently is how the replication itself may mechanically shape the 3D genome during S phase and during G2. I mean, most of the work that have been done is to try to understand the organization is in G1, mostly in G1. There is some works by Anton and others to try to understand what's happening in G2 and M. Very few try to address the role of the different mechanisms that can possibly act during the S phase. And so that's the question we want to address. And actually, in the past, there were a few hypotheses that leads about mechanisms that may impact the 3D organization via the replication. One is related to interaction between forks. So basically, when the replication initiates, there is two forks that will walk in opposite direction along the genome and that will start the replication and make the two sister chromatid. And one hypothesis is that potentially these two forks remain linked together in the 3D space. Potentially the act of replication may act as a loop extrusion mechanism for the two sister chromatids. And there is potential molecular evidence for that. And the other hypothesis that we'll not discuss too much today is also potentially the capacity to these forks from originating from different origin of replication to also interact in space via attractive interaction, for example, and that could form potentially these large-scale replication factories. And another crucial implication of replication is also because it prepares the two sister chromatid potentially to mitosis. Okay, so after replication, you have two sister chromatid, and you may have the question how the replication organize this sister chromatid one to each other in relation potentially to G2 and M phase. So the first thing we have to do is try to build a model that will couple replication and 3D organization. And to do that, of course, you need two ingredients. One is to account for the replication dynamics. And this is a very basic schemes of how people usually model replication. So you have factors that can bind to origins. So there is specific origins where factors can bind stochastically. And this will stochastically fire replication bubbles that is made of two forks that will walk in opposite directions. And two bubbles may meet each other and merge, releasing factors and creating a larger bubbles. And you may have also a replication of the ends that create this type of structure. And so to do that, we use the expertise of Jean-Michel, who has worked on this 1D models of replication along the genome for many years. And so we end up with an explicit model that can recapitulate the timing of replication as a function of time. So here this is in yeast. So all my talks, the application will be in yeast because it's a much simpler organism with less perturbing mechanisms occurring at the same time. And here is the prediction of the model, and here this is the experimental behavior for the mean replication time as a function of the genomic position. And you can see that this is very heterogeneous, very peaky around the genome. So there is regions where you have where replication is very early, and that's close to the origin of replications. And you have regions where it is very late, and this is basically the regions that are in between two origins of replication. Okay, and so the second step that we have to do is to explicitly account for the replication of the polymer. And so with that, we build an explicit model where we account for the duplication of monomers. So basically you form and you initiate a replication bubbles, and then forks can progress in opposite direction along the genome. And when they move from one bit to another, we will add another monomer in the system connected to the others, creating bubbles like that. And these bubbles can progress along the genome as the replication runs, and then you may have merging of bubbles. All right, so we have our kind of polymer model that is coupling replication and the 3D organization. And with this, we will ask two questions. First, we will focus on the S phase to try to understand how the act of replicating have an impact on the genome organization. And then we will address the role of sister chromatid cohesion. Let's start with the first part. So to characterize somehow systematically what's happening in the system, we first look at one replication bubble here that is dynamically growing as a function of time. And then we focus on the distance between the two forks, basically the two ends of the bubbles here, the green beads here. And so this is this quantity, so the average distance between the two, as a function of the genomic, the size of the bubble, more or less. And so something we can play with in the model is the speed of replication. Okay, so we can go from physiological speeds to much higher speeds or slower speeds. Okay. So what we observe is that basically at the physiological speeds, which are the blue line here, so this is the distance as a function of the size of the bubble, the dynamics of the structure, of the bubbles really recapitulate equilibrium ring polymer statistics. So that's the red point here. So basically the speed of the polymerase in vivo is very slow and allows the replication bubble to equilibrate as a loop. And so it follows that. When you increase the speed of replication by a factor 10, for example, that the green line here, you see that for small bubbles, you are still close to the equilibrium. But as the bubble size increase, then the dynamics of equilibration of the bubble is much slower. And it's slower than the progression of the forks, and then you start to deviate from equilibrium. And actually, you tend to converge to the initial linear like equilibrium properties. So this is the behavior of loop in this hypothesis where there is no specific interactions between the two forks. And when we introduce this loop extrusion-like mechanism where the forks remain together in space, of course, then the distance between the two forks remain constant. Okay, so no surprise here. What is more interesting and that also can help discriminate the two type of scenarios, so no interactions versus interactions, is to look at the global organization of this object. Okay, basically this object has many bubbles that are replicating at the same times. And so you can, for example, look at the radius of gyration of this object. And in this hypothesis where there is no interactions and you only have loops that are growing as a function of time, you have a slight decrease of the radius of gyration, and the minimum corresponds to the time where the bubbles are maximal, just before the bubbles merge together, so more or less in that situation. And we do see a more dramatic effect when we impose the interactions between the sister forks. So here, when we see that the decrease in compaction is much higher. And when we look at the anisotropy factor, which is a measure of how elongated, relatively elongated is the molecule, we do see again a peak of anisotropy in this situation where you have interaction between sister forks. And that's actually quite reminiscent to what is also observed in classical loop extrusion mechanisms mediated by cohesin, where you have the formation of kind of polymer brush that evolve as a function of the S phase. And that basically reach a climax somehow at the very end of replication. Okay, so we have very basic prediction about what's going on in terms of the structure. And so we wanted to compare to experimental data that are available that may potentially challenge our prediction. One of the experimental data, one of the few experimental data that exists is from this paper, that actually this paper was one of the key papers to drive this hypothesis of loop extrusion-like, even if it was not formulated like that in the paper, but interactions between sister forks. So in this paper, basically, they are looking at the distance between two loci that are symmetrically positioned around an origin of replication. So in the yellow beads is an origin of replication, and here is the two loci, okay? And they look at the distance between them as a function of replication, the function of time. And so basically we do the same for these two scenarios. So the first scenario is when there is no interaction between the sister forks, and the time of replication here is here. So basically you see that as the bubble, at this time, basically the bubbles will just start at the origin of replication, and you will have more or less no effect on the relative distance between the two loci. Okay, this is the same distance as you will observe in a kind of linear polymer topology. And as the replication progresses, you see a decrease in the relative distance between the two loci, and the minimum consists at a time where basically the forks are exactly on top of the two loci. Okay, so basically when you have a bubbles that is encompassing the two loci fully, and the two loci are the borders of the bubbles, and you have a decrease because a loop is a more compact object as a linear polymer, and so you have a decrease in the relative distance between the two objects, and then when the bubbles progress more along the genome, you start to have an increase of the distance. Okay. When you now impose the interaction between the two sister forks, the effect is the same, but it's much more dramatic, because when these two loci are replicated around the same time, they will be maintained together by this interaction between the sister forks. So then we can compare with what was measured experimentally using live microscopy. And you do see here, okay, even the experimental statistic is not very good, that actually this hypothesis of loop extrusion mediated by replication is consistent with this one single experiment. Of course, this is one experiment for just one pair of loci, and so we wanted to know if we can improve the statistics of this kind of comparison with our prediction at a much more genome-wide scale. And so for that, we thought about using Hi-C, okay, which usually is not used to address that question, but we have tried to dig into the data if we can see a signature of this event. So what the model can predict is that, okay, around origin of replication, okay, you have initiation of the bubbles, and since the two forks remain close to each other, this will correspond somehow to a dot in a kind of virtual Hi-C map. And as replication progresses, this dot will move perpendicular to the main diagonal because you maintain the interaction between the sister forks. Okay? And in the current chromatin literatures, for those who follow, this was also associated with what is called chromatin fontaines or chromatin jets, where you basically have loading site for cohesin. And here it's a bit the same. You have a loading site for an extruder, which is not cohesin, which is the pairs of forks that are moving together. And so the model will predict that as a function of time, you will form this kind of fontaines around an origins that will propagate a little bit along the diagonal and then vanish as replication ends. And so we basically took Hi-C data that hopefully and luckily were measured during S phase. Okay, and actually this is a collaboration with a friend in my institute. And what we do see, and when we look at the Hi-C average high-C-map around origin of replication is that in G1, you don't see anything. That's expected. And in early S phase, around origin, where the signal should be very strong, you do see this kind of flames or fountains that disappear in G2. Okay, so that's really a strong signature of this effect. Hopelessly, in yeast, at the same time that replication occurs, you also start to load coenzyme on chromatin, and as we know, coenzyme may also do loop extrusion. Okay, so we are not sure of what we are doing is actually a signature of the replication itself or a signature of coenzyme that is loaded basically also potentially at origin of replication, and it is doing the same job. So the first thing that we do is that first we use a mutant where replication is not there. Okay, you still have loading of coenzyme, but there is no replication anymore in the system. And so you lose the fountains. And the last experiment that we did is when you allow the replication to occur, but you deplete in coenzyme. So you should not have this confounding effect. And then you nicely recover these beautiful fountains that are actually stronger than in wild types, potentially because you remove the crosstalk with coenzyme. Okay, so rapidly, a summary of this first part, and I will go very fast for the second one. So we do introduce a new object, which is this self-duplicating chain that have very interesting physical features. And we show that this model can make very precise prediction on how the contact may happen, and that we prove experimentally, at least proof, I mean, we show that this is very consistent with high-C data. And very rapidly, I would like to go through the second part in the few minutes that I have. So in yeast, there is not really a G2 phase, it's G2M, so the G2 phase is very short, and you rapidly go to the M phase. And in yeast, you have, at the same time, as I said, loading of cohesion that will perform loop extrusion. So you have this loop extrusion that may happen, but you also have during the S phase the loading of cohesive coenzyme. Okay, that was the original role of coenzyme somehow, that to maintain the two sister chromatids together. Okay, so you have the two type of object, and so we wonder what is the coupling between replication, loop extrusion, and cohesion more in this system. And so in a high-C map, actually, what is interesting is that in the G2M phase in yeast, you do see these dots that are appearing that are not there in G1. Okay, you do see these dots that are the signature of contact. Okay, between regions that are named CARs for coenzyme-associated regions that are enriched in coenzyme that corresponds to usually to convergence genes. But anyway, so you do see these loops, okay, between CAR regions, and these loops may emanate either from interaction by loop extruding coenzyme, but also potentially by interactions between cohesive coenzyme, okay, that are bridging together two parts of the coenzyme. Because high-C map, or micro-C here, is blind of the sister chromatid identity. Okay, it doesn't know if this is sister chromatid 1 or sister chromatid 2. It just accumulates all the contact together. Okay, so that's a picture here. When you do see a contact here, it's potentially a contact between the loci on the same chromatid or contact between two chromatids. And luckily, there is Job Decker developed a technique that he applied in yeast that allows to potentially disentangle the interactions between the sister chromatids and within the sister chromatid. And when they do that, they still observe loops, but potentially loops at different positions between the two places. So meaning that the two sister chromatids are not perfectly aligned, but there is some dynamics between them. And so we wonder if we can make sense of this. And so I will skip that. And so the first thing we have to do is to try to build a model that can both integrate the cohesion and the loop extrusion. So first we start with the loop extrusion mechanism, and we basically try to fit what could be the density of loop extruders, the density of cohesin, density of barriers that are necessary to recapitulate what is observed inside one sister chromatid. So basically to describe the intra-chromatid contact. Okay, and so very easy to do that. I mean, very easy. It's possible to do that. And basically, these are the numbers that we get. So basically, it leads to an average loop density of 85 kilobase per and a density of barriers of about 20 kilobase per. Now we introduce the cohesion. And the cohesion in the data is basically described by this contact between the sister chromatid that is observed. Okay? And so when you do see loops between these two chromatids, actually there is two different possibilities. Either you see a loop because there is a cohesion between the two sites I and J here, and you have a loop between 1 and 2 inside one chromatid. And so you will see a contact between locus 2 of chromatid 1 and locus 1 of chromatid 2 because this is mediated by these loop extrusion loops. Okay? It's not because you have a real loops or a real contact between 1 and 2. It's just because you have contact between 1 and 2 intra and 1 and 1 inter. Okay? That's one possibility. So we call it you have symmetrical cohesion. So cohesion between site 1 and site 1, but you may have loops 1 and 2. And of course, you may have also these loops if there is a direct interaction between 1 and 2. And we call it the asymmetric cohesion loops. So I will not go too much into the details, but what we can show is that with the two type of hypothesis, you can nicely fit the average contact probability as a function of the genomic distance both intra and inter. So both models are compatible with that, and we have a very nice fit. But there is a prediction that really helps to potentially disentangle between the two. So what we have here is the average Hi-C map around a car, so around a site where there is a lot of cohesins. Okay? So either loop extruding cohesins or cohesive cohesin, we don't know. That's the experimental data for the intra-sister chromatid contact around a car. And you do see that you have this kind of interactions, but around the cars in the middle, it's quite depleted in interaction. Okay? But this is the intra, sorry. And this is compatible with loop extrusion-like mechanism. Okay, so that's the prediction of the simulation in the different scenarios. Intra is very well reproduced by the data. However, in the symmetric cohesion, so basically when you have cohesion only between sites that are positioned at the same place, the model will predict that in the inter-contact map, you will have a lot of contact very close to the cars because it's a symmetric cohesion. Okay, so the cars in the sister chromatid 1 will interact with the cars in sister chromatid 2. That's exactly the same one. And so they will have a lot of contact here and much weaker contact around. And actually in the data, you don't really see that. You see much stronger contact beyond the car. And actually the second model where you have asymmetric cohesion, so basically where the cohesion is likely or mostly between sites that are not aligned, but that are a little bit shifted. Now we recapitulate both the inter contact around the cohesin-associated domains and the intra. Okay, so just to summarize, in this very short and too fast part, we show that cohesion is very important for chromosome organization, and it may crosstalk with loop extrusions to organize the genome during G2M. So in yeast, of course. In mammals, I think it's quite interesting, and maybe Anton will talk about it later. But what is interesting is that the data, at least in yeast, are more compatible with this asymmetrical cohesion around the cohesin-associated domains. And of course, what we would like to now couple is really the full description of the replication coupled to the loading of cohesion and cohesive cohesin and loop exclusion cohesin in order to really see the full coupling between the replication and the cohesion. And this is to thank the people, and I thank you for your attention.